good afternoon, Professor Pereira, and thank you for finding the time for this short interview. In our continuous effort for communicating what we do in our cost action, we try to have some words from either the participants or the professors present at the different conferences. And today we want to delve more into your protein universe. And we would like to know more about what do you mean for the dark universe of protein and how these relate considering globular proteins, but also non-globular proteins.
Yeah. So the goal of this project or modeling the protein universe is really to try to help life scientists find what are these set of proteins that are poorly studied. And in our first attempt, we only looked at what would be like easy to look at. So really just the tip of the iceberg. And in that case, what we really did was to put a set of thresholds that ended up to kick out most of the proteins that would be an interest for the non-globular proteins community. So everything that is modeled with a low confidence by AlphaFold was eliminated from our first attempt. But we already with that, we were able to find a diverse set of proteins of unknown function, especially from bacteria. These are mostly, of course, globular proteins, but we also found some highly repetitive proteins. So in this case, they are not so globular, but that they are not annotated with known domains or known sequence features. But what this work brought was at least, of course, it helped us find new protein families in globular proteins and some repetitive proteins, but it also, more importantly, allowed us to set up these workflow to understand protein diversity. So now we can expand this workflow to all proteins, and then with this case, then start including also disorder in there, which I think is going to also bring a lot of darkness, but also brings a lot of challenges because their sequence features are different from globular ones, which is going to difficult some aspects of modeling the landscape, but it's going to bring us, I think, new interesting lines of research for the future. Yeah,
definitely. In your experience, or at least in your past experience, is there any specific example where your work has led to the identification of novel functions or interactions for what concerns globular proteins?
Yes, definitely. We found particularly, so in our first work, we bumped into a new putative toxin from bacteria, and this then, in collaboration, we validated that it's indeed a toxin and found also the antidote, which was also unknown, also dark. So with that, we discovered a new toxin-antitoxin system, which led us to a new direction of research of trying to find more systematically these systems, and we've predicted a variety of them, and also this led to the validation of another one. So in that realm of toxin-antitoxin systems from bacteria, we found a lot of diverse things. We also found a new predicted fold, which we call the beta flowers. And these are globular, but they are also highly repetitive proteins that can adopt a variety of shapes. But these we are still experimentally characterizing. We still don't know what it does. But these are also a challenge because the methods that are now available to predict function do not give us good predictions. So it also highlights that we have to work further in developing methods to predict function for these very hard cases.
Yeah, about the methods that are available around for prediction in general, all the methods that are either machine learning based or that are based on large language models, the speed at which these methods are evolving and developing, it seems incredibly quick. And if we were considering them as a black box 5 years ago, now the feeling is that also the scientists which are using them, they don't even know how they are working and how to interpret the process. Most of the times, in the best case scenario, they know how to interpret the results. And this is also, I think, sometimes even more overwhelming from the fact that there are also big companies working on this kind of algorithms, like Google or Meta. The question from your point of view would be, how would you advise young scientists to stay on track and don't get obsolete in this general landscape?
Yeah, this is a very difficult question because yes, indeed, the field is evolving really, really fast, and almost every day there's new methods coming in, and how do we keep track of all of this stuff? There's many groups that are heavily involved in benchmarking and testing different methods for different tasks. And so keeping track of those works, I think it's a good start. So look at papers, at groups that are testing these methods for different tasks and see which ones score the best and go with that. If you cannot do these benchmarks yourself. If you can, then always try to test a variety of methods for the task that you are working on, and then see which one is performing the best. When it comes to companies coming to the field, honestly, I think it's good for us to be aware that there's only so much we can do, that we cannot compete with what these companies are doing. But what I say to myself and feel that at least the people around me feel the same is that we cannot compete in terms of resources, but we have, at the end of the day, we have different goals. And what drives me in my research is really the biological question, and not necessarily come up with a new awesome language model. So if these companies are developing these methods and they know how to do it and they do it well and they have the resources, I'm happy for them for that, because then if I can use it for my own biological question, then it's good. So I always try to, in my research, to have a very specific biological question that will help the people around me. And then try to develop what is necessary to tackle that question, either by using resources that are already available, like the ones like AlphaFold, right? Or ESM, if ESM is the best language model for the task. But then use them, like build pipelines and workflows that make the best use of these resources for the particular question that we have, if that makes sense.
I mean, you are sort of saying that it's nice to outsource the power of companies. Mm-hmm. And at the same time, if the company can afford to spend time on something, first, it means it's relevant in the field. And second, it's not better not to compete, but you would rather count on their effort rather than trying to...
Yeah, it's not blindly counting that they are doing that. It's more of, if someone is already doing it, why am I even trying to do it if I cannot do it better? Mm-hmm. So let them do it, then I will use it for a particular question that is of interest to me and the people around me that the company necessarily is not interested in, right? So it's kind of like as if someone is building a car. Why will I try to build a car if they have the resources to do it? Then I can use the car to drive to the beach, you know? Mm-hmm. So, yeah.
Makes sense. About the finding the scientific hypotheses that are relevant, you recently set up your own lab at the VIB in Leuven, Belgium. And actually, I think from the outside, at least, you look like a very young PI. So it might be useful to give suggestions to young researchers that are trying to reach the same position or they are aiming to set up their own lab. What would be your take on that?
Yeah, from academically, I think when I started setting up my lab, I was already quite old. It was about 8 years after I graduated, so I had 2 postdocs, so quite of a senior postdoc when I did it. But even there, it was really scary because before you are always under the hood of someone that is, responsible, and you don't need to worry about. Well, you still need to worry, but there's things that are not your responsibility anymore. And so then when you want to give this step of now having your own group, it's a lot of responsibility. But when I was searching and looking for where to go and applying, I was always applying to the places where I really felt that this could be a good environment for me to work on, either because I know some people there and these people are happy, or there's groups that are doing research that is really interesting for me, or very complementary, let's say. Because I'm a biochemist by training and do computational biology. So I always wanted to work and develop. So when developing things, I always wanted to be close to the biological application as well. So with having someone around that it's working on very interesting biological questions that excite me. So that was like the first criteria when applying. And then when going for the interviews, always try to really make it. So this is the first part was about how to select where to apply or be criterious about it. But then when you reach the interview stage, this kind of connection is also very important. So if you applied, it's because you would like to be there, and you should definitely show that to the people that are there. Always try to, even when you're giving a shock talk, when you're giving a seminar, or when you're talking with people, really show that you are the right fit for that. Not just because you're trying to be phony, but really show your enthusiasm and have ideas of projects that you could work on. So already think about these things when you go for the interview. And basically show which kind of person really you would be as a colleague. Don't fake it. Just be yourself, because otherwise people will notice that you're faking it and will not like it.
Okay. You would go first for the environment.
Yes.
For I would say also complementary environment, but also you would go for somewhere where the connections with the people around are valuable in terms of human.
Exactly. Yeah, you should arrive there and be happy to go to work, because you're going to work with these people and you're going to have meetings with these people about boring stuff, and it should be nice, right? You shouldn't be just rolling your eyes that, oh God, I will. No, it should be exciting to go to work and be around the people that you just met. I think that's one of the most important things. And of course, then it comes with all of the other things of is this country good in terms of for financing your work? Are there good opportunities for funding acquisition? Could you easily collaborate with companies if it's something that is interesting for you? So I think it's good to do all of these research before, even before applying. But if you start seeing that you do all of these research and you don't find anywhere to apply, well, maybe you should reduce your criteria. But it's, I think it's very important to basically see, is this an environment that is good for the kind of research that I want to do, but also for the kind of life that I want to have, being it work-wise, but of course, person-wise, personally. The people the institute where you're going to work, the people that you're going to see every day, they're also going to define how your life is going to be, and you should be happy and feel happy about this prospect.
Yeah, definitely. I think a lot of scientists will keep an eye on your work, both in the globular protein field and also non-globular. And I really hope to see more of your work in the next future. So we wish you, as a cost action, we wish you good luck for your next years. And thank you for the interview again.
Thank you.